guys, good morning. So what I'm going to do today is we're going to be talking about biomolecules. And so with biomolecules, we get all of our nutrition and our energy from the foods that we eat. And so to kind of demonstrate this, I'm going to go through what I normally have for breakfast on a school day. So I just got back from walking my dog, got her breakfast. Now it's time for me to eat breakfast. And so I'm going to show you what it is that I normally eat. So let's get started. Okay, so I already have my oatmeal. Okay, so I know that's an old person breakfast. It was something that I picked up when I lived up north, just when you want something that's nice and hot, but it doesn't take too long. And so I have oatmeal over here, boiling up, and then I have a couple of things here in this bowl. So I have some sliced up banana and I have some peanut butter. So let's go ahead and look at those. So I have the containers right back here. Okay, so I don't know if you could see this, but if you look at the back of my oatmeal, you could probably see that, there's some fats here. Let's see here, so some cholesterol, so thankfully that's 0. And then I have some carbs here. Okay, so the things that are highlighted are fats, carbs, and proteins. So I can see that my oatmeal has, quite a few carbs, which makes sense because it's a grain, and then it has a little bit of protein. So next, I like to use almond milk from AP. Okay, so same thing. Okay, so I have fats, I have carbs, and I have some proteins. And so, whenever I'm looking at all of these foods, even my peanut butter that I have in my bowl, I can see it's the same thing: total fats, total carbs, and total proteins. And so the reason for all of that is because those are the 3 main biomolecules that we need in order to build up tissues and provide our cells, good functioning for the body. So I'm going to go ahead and finish this up, and then we'll go ahead and move on covering those 3 different types of biomolecules. Hey guys, so I'm back. So what we're going to do today, we're going to go ahead and talk about our biomolecules. So hopefully you enjoyed my little impromptu cooking show and you got to see how, we can get some of those biomolecules. And now we're going to learn how we use them and why we actually need all of those different types. So let's go ahead and get started. And my mouse does not want to show up. There we go. All right, so we're going to talk about biochemistry, and we're also going to talk a little bit about our properties of water. So let's start with water. So water is going to be our universal solvent. It's an inorganic compound, and all the different properties allow us, to be able to exist. So for example, water has hydrogen bonding, and so that allows for different compounds, to be able to travel throughout our body. It allows for cell transport. It even allows for stuff that we don't think about. So like if you ate, like let's say a hamburger that you left out on the counter overnight, maybe it's not good. There's some extra bacteria there. The water is going to help dilute those bacteria, and it kind of helps, protect you. And so, because of water, water allows our cells to function in the ways that they need to. Additionally, in the environment, if we're looking at the water structure, whenever it freezes, it floats. And so that also creates another environmental niche, for certain animals. Additionally, let's go ahead and talk about carbon atoms. So carbon atoms are super important. Anything that is organic means that the basis of it is going to be made out of a carbon atom. And so the reason why carbon atoms are so special is if you look at their structure, they have 4 valence electrons, so 4 free electrons that are floating around that outer shell. And so it can form up to 4 covalent bonds. Covalent bonds are pretty sturdy. It helps create our different biomolecules. And then you can also form single, double, and triple bonds. In addition to all of this, you can have so many different shapes and structures. You can make them as long or as short as you want. So like ethane and propane, so having 2 carbons in the middle or 3 carbons in the middle, you can have hundreds of carbons in the middle. And so, those will help us make different types of biomolecules. You can also insert double bonds in different positions. So here's one at the end and here's one at the middle. You can also branch them. So you can have multiple types of branching carbons, and then you can also have different rings. Those of you that have to take organic chemistry whenever you're in college, you're going to be seeing a lot of these rings because it's really important for organic chemistry. So not only can we have our carbon skeleton, we can also add on functional groups. So I'm not going to go over all of these functional groups. I'm just going to go over some of the important ones. So for example, up here, we have our hydroxyl group. So hydroxyl is characterized by having an OH. Anytime you have that OH, it symbolizes an alcohol. So you could have ethanol, propanol, butanol. Anywhere, if there's an OH on there, it's going to be an alcohol.
So I'm going to skip on down here to my amino group. Amino group is my NH2. So that may sound familiar from amino acids. We're going to talk about those in a little bit. But if you think about an amino acid, that means that there's an amine group there. Last one I'm going to talk about is down here, my phosphate group. So hopefully you saw my video from yesterday and I talked about ATP and phosphorylation. Phosphorylation is when you're adding an inorganic phosphate group. Inorganic means that it's not attached to a carbon. If you look right here, this says it's an organic phosphate group. And so if I look at this right here, there's a carbon attached right to this phosphate group. But phosphate groups are important. It's a very large source of energy. So I can look at these two molecules. So I have estradiol and testosterone. Those are fairly common hormones, and they look a little bit similar, but their structures are just slightly different. And they're different enough that they're going to cause different effects on the human body. So for example, if I look right here, if I have two lines, that symbolizes there's a double bond. I can see that this one ring over here has 3 double bonds, while the one in testosterone only has 1 double bond. Additionally, I have different functional groups, so these two are the same, but the ones in blue are all different. And so that's going to allow those molecules to function just a little bit differently, which is important for how we function and maintain homeostasis. All right, so let's go ahead and talk about macromolecules. So macromolecules are polymers. Polymers are very large structures of carbon skeletons put together. So if you look at that little camera that's down at the bottom, you're looking at me. You're seeing all of my macromolecules, my skin, my hair, my eyes. All of that is a type of macromolecule. And so we're going to talk a little bit how we're building and breaking down those macromolecules. So a very large macromolecule is known as a polymer. So a polymer is just a string of monomers put together. So a monomer is the building block. And so if I look right here at my illustration, my monomer, this tiny little Mickey Mouse, is a small molecule. It's the most basic I can get when we're talking about a biomolecule. If I add a bunch of monomers together, that's how I create my polymer. And polymers can be big and they can be small. So how does this happen? How do we do this? We have two reactions, one called dehydration synthesis and the other called hydrolysis. It's exactly like it sounds. So if you're going through dehydration synthesis, you're going to dehydrate, you're going to pull water out and synthesize two molecules together. So you're going to be able to make a bigger molecule by pulling out the water. Hydrolysis is the opposite of that. Hydro is water and lysis means to break. You break apart that water and stick those molecules back on, and you're able to break apart that polymer because now you've added on those extra atoms. So let's go ahead and talk about some of those biomolecules and figure out why it's important. So first we'll talk about our energy molecules, our carbohydrates and our lipids. This is the stuff that we like to eat. So like there's a picture of a hamburger right here with cheese. We all love that, okay? So we love having carbs, as well as having lipids. And I know this is talking about a low carb diet, but we can find out that there's some other things on here. So like for example, lettuce and tomato that are also carbs. So generally speaking, when we're talking about carbs, because that's what we'll start with right now, we're talking about sugars and starches. So sugars and starches are made out of carbon, hydrogen, and oxygen, and it's always in a 1 to 2 to 1 ratio. 1 carbon, 2 hydrogens, 1 oxygen. And the primary function of carbohydrates is to provide us with short-term energy. So if you're ever feeling sluggish, you're feeling a little slow, or maybe right before lunchtime, and you eat something, your body is going to immediately use those sugars from whatever you ate to kind of pep you up. The problem is if you have too much sugar, and you're not supplementing it with proteins or lipids, you could have a sugar crash because your body uses all of that pretty easily. We can also use it for some raw materials as well as structural materials, and we'll talk about that here in a second. So some common sugars, monosaccharides and disaccharides. So mono means one, that's our monomer. So those are things like glucose, galactose, and fructose. Those are our simple sugars. Disaccharides is when you have 2 monomers put together. So that's things like maltose, sucrose, and lactose. And you can even get more complex than that. So if we're talking about energy storage, so for example, what plants need to do, they're going to do this by producing starch. So starch is a very long polymer. And we'll look at what that looks like in a second. And then we also have glycogen. That's what we store up in our liver and our muscles. That's one of our last, defenses as far as, if we need to break down sugar for anything like, a fight or flight, we're going to be, harvesting that glycogen that's in our liver. You can also use it for structure, such as for cellulose and chitin. And so those will help, plants, for example, create that firm structure. And that's why when you bite into celery or carrots, it makes that crunching sound. And then chitin is actually what gives insects that really firm shell. So if you step on a bug and it crunches, it's because you broke apart that chitin. So if we're looking at starch versus glycogen, you can see that you have these long rings all attached together. And so starch is pretty easy for us to digest. And so, this ring, you can see it's pretty simple. They're all attached to just one. They're not really branching. It's all linear. And so for starch for us, that's stuff found in like corn and potato chips. And so it's stuff that we do like to eat. But for a plant, it's going to be their storage molecule. And that's just because, it's so long, they're able to store all of those carbs in that long chain. For us, glycogen is very similar, but it's branched. And that's because we don't use glycogen all that often. We only use it, in emergency situations. But because it's branched, that means that these little rings can break off in several different areas for a fast release of energy. And so in fight or flight, you're going to want to have energy really quickly. And so those little rings will break off very fast. So next, let's go ahead and talk about lipids. So lipids is another type of energy storage, but this one's a long-term energy storage. Again, composed of carbon, hydrogen, and oxygen, but in no particular ratio. We have three major groups for lipids. We have fats, we have phospholipids, and we have steroids. So the thing with lipids is that you don't get, polymers, you just get this continuous branch, of these carbons and hydrogens, and that's what makes them really, really long or really, really dense. So the only fat that I really want to talk about, the only lipid I want to talk about is a phospholipid. So for a phospholipid, this is part of our cell membrane, and we'll talk about cell membranes when I talk about cell transport. But it's really important to remember for our phospholipid, it has a hydrophobic tail and a hydrophilic head. That makes it amphipathic. And so that's really important with our cell membrane because it allows only certain things in or out of our cell membrane. It's not going to just be, permeable where everything can go in. And so it's going to help protect us and allow us to get what we need inside of our cells and keep the dangerous stuff out. Okay, so I just explained why that was important. So steroids are also our 4 fused carbon rings that are all together, and then you add on your different functional groups to make them, different. So for example, cholesterol is a steroid. I showed you testosterone and estradiol. Those are also steroids. All right, so proteins. So for proteins are made out of amino acids. Amino acids are going to be my monomers that make up those bigger structures of proteins. So proteins are extremely versatile. They make up pretty much everything in our body. And also, if you're looking at any other living thing, they're made out of proteins. So if you're looking at amino acid, there's my amine group. That should look familiar. I have an R group with my central carbon, a hydrogen, and then I have a carboxyl group. That's one of my functional groups as well. And so this is what makes an amino acid. So you can attach those all together in different ways and form these peptide bonds that are in the middle. And these peptide bonds will allow for a larger protein. And so if you add on so many amino acids, then you're going to get varying types of protein. And then not only that, they're going to fold and they're going to twist and they're going to bond in different ways. And that's how you get your different structures. So just to review really quick, so you can have just your string of amino acids and that's your primary structure, just amino acids all stuck together. Next, you can have your alpha helix or your beta plated sheet. So you're forming bonds to either create that helix shape or that sheet, that folded sheet shape, and those are formed by hydrogen bonds. Our tertiary structure, you're going to have hydrophobic interactions and disulfide bridges. So if you get a bunch of these alpha helixes and beta plated sheets and you put them together, that makes a tertiary structure. And then lastly, let me move my little camera. My quaternary structure is a bunch of tertiary structures put together. So remember, to denature a protein, it's the same thing as denaturing an enzyme. You change the pH, you add in salt, you change the temperature. It's going to disrupt those bonds and make them reform in different formats. And so you're going to change the shape of that protein. So last, nucleic acids. So nucleic acids are very, very simple. The only purpose for these is to store and transmit hereditary information. So you have 2 types. You have DNA and you have RNA. And both of those are made up of monomers called nucleotides. So if we're looking at a nucleotide is going to be my nitrogenous base, which are my carbon rings that are in the middle, my sugar, and then my phosphate group. And so this right here, this little green pentagon and my little yellow circle, that makes up the backbone of my nucleic acid. And then these in here are what gives us our genetic information. Our nitrogenous bases give the genetic information. So our nitrogenous bases are connected together with hydrogen bonds. A always goes with T, G always goes with C when we're talking about DNA. So these bases have to match because those hydrogen bonds have to match together. And not only that, when those strands separate and they go through replication, they need to be able to replicate accurately. All right, so that was it. That was a really quick overview of biomolecules. Hopefully I didn't go too fast. If I did, go back through, rewind, and rewatch. So that was just the basics for biomolecules. If you have more specific questions, make sure you email me and I can make sure to answer. All right, I hope you all have a great day. Bye.